Hi, welcome to the Pelago webinar, Navigate Your Chemistry and Explore Your Biology using CETSA. My name is Ylva Elias, and I will be presenting first here today, giving a short introduction. And then my colleagues Helena, Alexei, Benedict, and also Stina Lundgren will be presenting about the latest applications of CETSA. I also want to mention that you are all muted here, no videos, but please feel free to add your questions in the chat. And then in the end, we will have a Q&A session where Stina will help out answering questions. So please feel free to put down questions in the chat during the webinar. And welcome again. We are really happy to see so many with us here today. And I will start with introduction. So Pelago is the home of CETSA. We are a specialized CRO and we focus only on CETSA. We have the exclusive control of the CETSA IP, and we also have an in-house CETSA R&D team, which you will hear more from today. We're based in Sweden, just north of the Karolinska Institute in Stockholm. CETSA can be used all across the drug discovery pipeline. So far, we have around more than 300 projects, all stages at Pelago, and also with our clients. And it's all from target-based drug discovery to lead generation, lead optimization, into pre-clin and even clinical development. In the beginning, we ran more few compounds, but now we also have methods for hit confirmation, screening, and we also have a mass spec applications for target deconvolutions and other applications, mode of action studies, biomarker investigation. And today we will be focusing more on our screening and primary screening and profiling of degraders, and also on how CETSA has been used on therapeutic antibodies. CETSA stands for Cellular Thermal Shift Assay. It's used for the native cellular environment for measuring target engagement in the cell. This is different from other more traditional thermal shift assays that is more for purified proteins. With CETSA, this can be used in the cell for getting closer to biophysiological conditions. And the principle is, if your compound binds to the protein, there will be a shift in the melting behavior of the protein compared to the native protein. And here you see actually a stabilization event. So after the heat shock was applied, the protein melted later than compared to the native protein. But it can also be destabilization events. And these are also considered true target engagement events, and this could be for associated proteins and other more complex proteins downstream in the cellular pathway. But in both cases, stabilization or destabilization events are both true CETSA target engagement events. And the core protocol is to treat and then apply a heat shock, and then separate and detect. We measure the soluble proteins that are left in solution after the separation, after the heat shock. We have two different formats, which is the CETSA Navigate, that is a targeted CETSA for antibody-based detection. And then we have the CETSA Explore, which is the proteome-wide CETSA for mass spec-based detection. This is also known as TPP, thermal protein profiling, or PISA, protein integral solubility alteration. But here at Pelago, we call it CETSA Explore for the proteome-wide CETSA. And it can be used with a broad range of samples: human, animal, intact cells, lysate primary cells, PBMCs, solid tissue, xenograft bacteria, even zebrafish, you name it. And you're welcome to contact Pelago about targets, and we can figure out the CETSability and all wide range of cellular cell lines and also other kind of tissues. And that was my short introduction. And now I will actually hand over the floor to my colleague Helena, who will talk about CETSA Navigate HT, how it has been applied to primary screening on CDK4 and STING.
So thank you very much, Ylva, for that. As I said, my name is Helena Almquist, and I work as a senior scientist here at Pelago. And I will talk about the CETSA Navigate HT formats and how it has been applied in an internal development project. But first, as Ylva said, the CETSA formats share the same core protocol. Within the CETSA Navigate HT format, all steps are performed in plates, all from incubating cells with compounds to the heat treatment and to the lysis of cells using a lysis buffer, and also the detection step. And the detection is generally done using the alpha technology, seen in the right corner. And that is based on 2 primary antibodies detecting the target, and then they need to be in close proximity to get a signal in the end. So after an assay has been either developed, the first step is to do melt and shift curves using tool compounds, and that can be seen in the middle, that 2 compounds nicely stabilizing the target, comparing it to the native protein melt curve. And another step you can do is to do titration of compounds and ranking potencies of compounds. And then you select one single temperature where you screen your compounds at. What you also can do using this single temperature isothermal CETSA study is to do a single point concentration, single point study using a full library. So you can screen for novel chemistry to, for example, start a new lead program. And that is what I will talk more about today that we have done at Pelago. But first, I will introduce you to two success stories from 2 clients of Pelago. So first, Vertex screened a quite complicated target. It was a multi-protein complex, target challenging to drug. No small molecular binders were currently known for this target. So the assay, the CETSA HD assay, was developed at Pelago. And then the assay was transferred to Vertex, where they then screened 50,000 libraries screened on this target. And the CETSA screen delivered new chemical scaffolds not identified in other screens. And then another case where that AstraZeneca screened 500,000 compounds targeting CRAF. And the detection method was using the alpha-LISA Surefire Ultra Detection Kit that has been validated for CETSA at Pelago. And the outcome of that screen was that they confirmed known CRAF chemistry, but also they identified type 1 and type 2 and potential allosteric inhibitors, and also hits without known kinase structure. And this really shows the strength and the power of the CETSA technology, that you can identify different mode of action binders in the same screen, and also screen targets that has a history of being undruggable or difficult to screen. So to the case study at Pelago. So we also wanted to explore and understand more about the potential that primary screening can give using the CETSA method. So we did a target selection approach in-house, where we looked at both the biology and target validation of different targets. And we also looked at indication, how they were the clinical development stage for these targets, and also accessibility approach, and also the potential added value using the CETSA method. And we selected 2 targets, CDK4 and STING. CDK4 being a well-established oncotarget with already marketed drugs. But we thought that this was an interesting case that could provide maybe novel chemistry, but being more of a proof of concept target maybe. The other target is STING, stimulator of interferon genes, is a membrane-associated target. It's involved in DNA-driven immune response. No marketed drugs yet, but a very hot target in the community right now. So we thought here that it was a good opportunity to screen for new novel chemistry, and also the potential to screen for agonist and antagonist, because this target has been many different modulators that it's been reported for this target. The approach that we took was that we first developed both assays in 2 different cell lines. And then the detection was done using alpha-LISA Surefire Ultra Crits for Perkinelmer that we first validated in-house. And then the compound library selected, we screened 11,000 compounds for both targets using the same library as a counter screen against each other in a way. And the library consisted of both focus set and a lead-like set, and pressed with well-known annotated drugs, and also a fragment-like sub-library. The pre-screening activities was done to test the robustness of the assay using the automated workflow. We did Z prime factor calculations at different temperatures, and also we did screening temperature selection based on the results with tool compounds. In the primary screening then, we used pre-spotted plates, so the compound had acoustically dispensed into 3 to 4 well PCR plates, and we screened the compounds at a final concentration of 50 micromolar. Then following up the hits, we decided to actually follow up all the preliminary hits from both screens against both targets as an also a counter screen. And the reason why we decided to follow them up at 50, 10, and 2.5, I will talk a little bit about later. And then the next step, this is where we are today, that we are currently looking into the hits and we decide on follow-up studies. And we will do full concentration response curves in CETSA and also melt and shift curves and some functional studies. So this is a slide showing a little bit from the pre-screen data. And here, the first is CDK4, the melt and shift curves using 3 well-established tool compounds, palbociclib, ribociclib, and abemaciclib. And as you can see, they all nicely stabilize CDK4. And the 3 dotted lines just represents the 3 different screening temperatures that we tested. We decided to screen in THP1 cells for this target, and 46 degrees, being the dotted line in the middle, was selected as screening temperature. The Z prime factor was very good, 0.8 for the stabilizing assay window, and that is from the isothermal control and up to the control, 100% control. So for STING, we had different modulators, both stabilizers and destabilizers. And as you can see, this DABC STING, being a STING agonist, clearly stabilized STING, whereas the 2 covalent inhibitors, H151 and C178, is destabilizing STING. And K562 cells were selected for screening, and 51 degrees were selected as a good screening temperature, where we can have a good stabilizing, both a stabilizing and a destabilizing assay window to identify both types of modulators. And then moving over to the screening results, this figure shows the plate layout, that the compounds from the libraries were populated in column 1 to 20. And then in 21 and 22, we had the DMSO controls, just as an isothermal control. And then in column 23 and 24 were the 100% and the 0% controls. That is unheated cells and cells heated to 70 degrees, which is where the protein has melted. And then this scatter plots show the data from the entire screen on CDK4. And the dotted lines represent the average controls that are normalized. And then the colored compounds in the middle, or the colored dots in the middle, represents all the compounds applied. And as you can see, we clearly identified some stabilizing, nice stabilizing hits, or preliminary stabilizing hits, I would say. And the screen worked really well from a technical perspective. The Z prime factor was 0.8 over all plates. And in the upper right corner is an example plate from the screen. And you can clearly see some of the stabilizing hits. So STING, on the other hand, looked interestingly very, very different. That, as you can see here, that we identify some clear stabilizing hits, but we also identify a massive amount of destabilizing compounds. And again, remember that we screened the same library. So this is some target effect that we see on STING. So again, the screen worked well from a technical perspective. We had a good Z prime factors for both the stabilizing window and the destabilizing window. And again, the example plates up in the right corner shows an example plate. And again, you see the couple of destabilizing hits, but also some stabilizing hits. So, and as I said before, that we continue to with the hit confirmation screening at 3 different concentrations, because we thought that at STING 50 micromolar maybe was a bit too high, since we got this massive amount of destabilizing hits. So we decided to confirm or to rescreen all hits from both screens in the same assays, in both assays. And this plot here to the left shows the correlation plot from the primary screen versus the hit confirmation screen on CDK4 at 50 micromolar. And as you can see, there's a very nice correlation of the hits, and we confirm over 90% of the hits. The vast majority of compounds here is actually STING hits, so they don't do anything on CDK4. They have no effect on CDK4. And to the right, you see a couple of examples of the stabilizing hits. So in black, you see that the concentration-dependent stabilization from 50, 10, and 2.5 micromolar on CDK4. And on STING, we actually see that they destabilize STING here. And palbociclib being the internal control, a nice potent concentration dependence stabilization of CDK4. On STING then, the same plot, the correlation plot from the primary screen versus the hit confirmation screen. We also see a very good correlation. Here is the stabilizing hits, and here are all the destabilizers that we confirm at 50 micromolar. But again, I said that we saw that 50 micromolar might have been a bit too high, but we did confirm many of the hits at 10 and 2.5 micromolar. And the hits here in the middle is probably compounds or that is active on CDK4 but has no effect on STING. On the right, you see some preliminary stabilizing compounds for STING with no effect on CDK4. And at the bottom, we see the diabsy agonist on STING that nicely stabilizes STING, and also the covalent inhibitor nicely destabilizing STING. So this is my last slide, just summarizing that we have done a successful primary screening campaign at Pelago on CDK4 and STING. We are at the moment doing clusteral and structural assessment, and we look at common structural motifs, potential assay interference among the compounds, and also other potential structural liabilities. And we are at the moment selecting compounds for further functional studies and also more CETSA studies to confirm the hits. With that, I will hand over the floor to my colleague Benedict, who will talk more about another CETSA Navigate format.
Thank you, Helena. So I'm Benedict von der Heide. I'm a research scientist, and I'm also part of the high throughput unit. But today I will show you data from our Western blot-based detection method that we're also going to use for hit confirmation, but we also looked at for a bit of the downstream biology of STING. And in order to do that, let's start by having a look at the CGAS STING DNA sensing pathway. So to begin with, there are several stimuli that can trigger the activity of cyclic GMP-AMP synthase, or rather abbreviated CGAS. And when CGAS is triggered, it will start synthesizing cyclic GMP-AMP, abbreviated CGAMP, which is also one of our tool compounds. So once CGAMP is synthesized, it binds to STING, upon which STING will relocalize from the ER to the Golgi and meet TBK1, for example, tank binding kinase 1. Upon that, TBK1 will autophosphorylate and in turn phosphorylate STING as well. And this triggers the downstream several downstream cascades. For example, one being phosphorylation of IRF3, which then in turn will result in a DNA-driven immune response. So we not only wanted to observe target engagement, but also part of the downstream biology. And this is nicely exemplified in the example by Chin and colleagues here. And when they profiled their compound, they could observe phospho-TBK1 after 30 minutes of compound incubation, and already after 1 hour, they could detect phosphorylated STING. So we are aiming to reproduce the same with our tool compounds. But before I show you the data, I'll give you a brief overview of the workflow. So first of all, the cells were harvested, then incubated with the compound for 1 hour at 37 degrees. So it's an intact cell experiment. The samples are then aliquoted, subjected to heat treatment at a temperature range, and then lysed by freeze thawing. The samples are then centrifuged, the soluble fraction is taken, and the Western blot is run. And then ultimately, by plotting the Western blot intensities, the melt and shift curves are generated. So let's have a look at the first tool compound being CGAMP. So the layout of the slides will be similar. So for the membranes, you will always see a compound incubated sample directly next to the vehicle control, and that for any given temperature. And in the quantification, you'll always see on the x-axis the temperature and on the y, the normalized Western blot intensities. So if we now have a look at the membrane, if we look at 53 degrees of the vehicle control, we see that we can't detect STING anymore on the, we cannot detect STING anymore on the membrane. But then if we compare to the compound incubated control, we can nicely observe a thermal stabilization of STING, which is nicely reflected here in the quantification. Then if we look at one of the interaction partners, TBK1, we see virtually no difference, which is also exemplified here by almost completely overlaying curves in the quantification and virtually almost the same melting temperature. So now going further downstream in the biology, where you just see really schematically and briefly sketched out the flow of what we expect. So first now we would expect to see phosphorylated TBK1. And having a look at the membrane now, we can see some cross-reactivity of the antibody, which is known from the supplier. So the main message here is that we see more phospho-TBK1 in the compound incubated sample as compared to the control. So seeing this, we also would expect STING to be phosphorylated, and that is exactly what we see, and it's exclusive to the compound incubated control. So we have not only observed the thermal stabilization of STING, but also could confirm the robust downstream biological response that is induced upon target engagement. So let's have at another tool compound that Helena talked already about as well, which is diapsy. Right here. So if we now look at the compound incubated samples, we see the appearance of a double band. And since this is quite hard to separate in the quantification, this band is treated as 1 in the graph, in the quantification, sorry. But if we now look again, start at 53 degrees, in the control sample, STING is not detectable anymore. But if we now compare to the compound incubated sample, we see that a massive thermal stabilization has been induced and we can detect STING even up to 63 degrees. If we now jump directly to phospho-STING, we see exactly what we expect. So all the data I have shown so far has been generated in K562 cells, but we wanted to compare this with THP1 cells as well. So having a first look at cGAMP, again, we see a nice compound-induced thermal stabilization of STING, and as expected, phospho-STING is present in the compound incubated sample. The same with diapsy. The double band appears here as well, but the message remains unchanged. We still observe thermal stabilization and phospho-STING is induced. So there is a lot of interesting observations that can be made from these experiments. And if you look closely, you might have seen one already. And this is one I'm going to share with you today, which is the melting temperature of STING depending on the cell type. So here in dark blue, you see the melting curve for STING in K562 cells. And in the brownish tone, you see the STING melt curve in THP1 cells. And you can observe there's a huge delta in the melting temperature of about 5 degrees, depending on the cell type. And so all these experiments have been conducted at least 2 independent biological replicates per cell type. And this is also something that has been observed in the HT format in the initial experiment. So then you might want to ask, is there an effect on, for example, TBK1 as well? And the answer is no. We see directly overlaying curves and literally the exact same melting temperature for both cell types. So while it appears that the melting temperature for STING is different between the cell types, it is not for TBK1. So to summarize and conclude, we observed target engagement of our tool compounds and replicated the finding in CETSA Navigate. we could observe a robust downstream response, and that was recorded by measuring phospho-TBK1 and phospho-STING in not only one, but two cell types, that was K562s and THP1s, and where it appeared that the melting temperature of STING showed cell type specific differences, while TBK1 actually remained unchanged. So in conclusion, we used CETSA Navigate for confirmation of the target engagement of the STING2 compounds, and also interrogated the corresponding downstream biological response that is triggered upon target engagement. And with this, I will hand over to my colleague, Alexei.
Thanks, Benedict. Hi, everyone. Thanks for joining us in this webinar. So now I'm going to switch gears and talk about CETSA Explore, application of CETSA to profile whole proteomes. So the combination of the CETSA principles and the power of MS-based proteomics allows one to measure compound-induced stabilization or destabilization of the virtually all proteins in the cells in one go. And it was first published in 2014 using isobaric labeling to facilitate quantitation of multiple proteins in one go. And in that paper, the feasibility was shown, identifying both stabilized and destabilized proteins in one experiment. These days, instead of focusing on measuring the individual protein melting curves and calculating the delta Tm to deduce the binding events, we measuring directly area under the protein melting curves. And comparing these areas under the melting curve, we can basically calculate the same, observe the same event of stabilization or destabilization. And we took staurosporin as an example, promiscuous kinase inhibitor, and we applied two methods. One classic method measuring the melting temperature and difference between them, and another one measuring the areas under the melting curves and calculating this delta S value. And what we found out that they correlate pretty well. And so basically, it doesn't matter what value to look at. And the good thing about this is that to measure the area under the melting curve, we don't need to measure the protein level at every single temperature point. What we need to do is just to pull all the samples directly after heating and then analyze one single sample to get the area under the protein melting curve. And this approach was tested and published in several papers and seems to be working really well and being very sensitive and robust approach. So in this method, which we call compressed CETSA MS, this value we measure, it's not only change in protein stability, but rather a combination, change in protein stability and change in protein abundance in the soluble fraction. And this gives us an extra dimension in our experiment to explore. Despite we used to keep the incubation time short in CETSA MS experiment, there are still some events which might affect the level of the protein in a sample within the incubation time. And then there could be several cases like this one to the left, shown when we only observe the change in the protein stability, which would result in a right shift or left shift of the melting curve. And then if we would measure the protein stability alone, we would see the signal. If we just measure the protein abundance difference, we won't see any signal. In compressed CETSA MS, we of course see the signal, but in sometimes when the protein abundance change, we won't see any change in thermal protein stability. But in a compressed CETSA experiment, we would still detect these events very easily. And this brings us to an idea to apply the power of CETSA to profile the novel modality of the chemicals, protein degraders. The reason for this is that in one experiment, we can measure both compound-induced changes in the thermal protein stability and compound-induced change in protein abundance. So we started with profiling thalidomide and analogous. And the reason for this was that 5 years ago, the mystery of the thalidomide, which can cause terrible side effects, teratogenity, this mystery was resolved, and it was found out that these compounds have very interesting mechanism of action. They bind to a single protein target in the cells, cereblon, which is part of the E3-ubiquitin ligase complex. And the binding of the molecule to the cereblon changed the substrate specificity of this E3-ubiquitin ligase, which leads to the subsequent degradation of novel IMiD-induced targets of this E3-ubiquitin ligase. And some of these targets been linked to the teratogenic effects, and some of these were linked to other effects, anti-myelomic effect, sedative effect, and others of the IMiDs. So we took the pomalidomide as an example, and we incubated pluripotent stem cells with pomalidomide, and then in one experiment we measured non-heated samples and the compressed CETSA samples, and we repeated this experiment at different incubation time. And by doing this, we were able to characterize the response in this two-dimensional area, when one dimension would correspond to the change in protein stability and another dimension change in protein abundance. And as we can see here, the cereblon is the only protein showing clear stabilization with almost no change in protein abundance. But if we look at the protein abundant change, we can directly see a number of proteins degraded upon the treatment with the thalidomide. And these are those proteins which were recently identified within the last 5 years as novel targets of the IMiDs. And of course, we could look at this in a time-resolved manner, and then we can see that the binding to the target happens in no time. So at the 15 minutes, we already see clear stabilization of the cereblon. But degradation of the now substrates started to happen already after 1 hour. And then we took it one step further and applied the CETSA MS to profile the product molecule. It's the chimeric molecule consisted of 2 parts, one of them recruiting E3-ubiquitin ligase and another one recruiting the protein of interest. And by doing this, it will cause to the ubiquitination and subsequent degradation of this protein of interest. So we took MZ1, recently developed product molecule, which is specific BRD4 degrader, and it works by recruiting the VHL protein as the E3-ubiquitin ligase. When we do the CETSA MS profiling, we can clearly identify VHL as the stabilized proteins and the BRD4 as clearly and specifically degraded protein in one experiment. And then we did another study in the light cells and in the intact cells with inhibited proteasome, and we could clearly see that inhibition of the proteasome reversed the degradation of BRD4. What we also see is not only BRD4 protein level goes down, but also BRD3 and BRD2. And for the BRD2 and BRD3, we see both compound-induced degradation, but also stabilization of the proteins upon the binding to the product molecule. And this, of course, goes in line with the previously published study. Yet another example, we apply CETSA MS to profile the product molecule, which consists of the pomalidomide as the E3 binding moiety and the palbociclib as the CDK4/6 specific binding moiety. And this product molecule is the specific CDK4/6 degrader. And in this experiment, we profiled palbociclib alone and the product molecule. And we've done this in both intact and lysed K562 cells. And if we look at the palbociclib results in lysed K562 cells, we can see very clear and specific binding profile of palbociclib, including stabilization of CDK6, stabilization of CDK4, some other off-target kinases being stabilized, and also destabilization of the PLK1 kinase. When we switch to the intact cells, when all the pathways are on, we can still see clear stabilization of CDK4 and CDK6 and off-target kinases. But what we also see is a destabilization of RB1, which is downstream of the CDK4 and CDK6 kinases. And usually it's being heavily phosphorylated by the CDKs. So we can see both direct binding events in intact cells, but also the downstream events in the cells. When we look at the product molecule, then the binding profile in the lyse cell is quite similar to the palbociclib, with addition of the cereblon as the target of the homonidomide part of the molecule, but also some more proteins specific to the product molecules, but still quite specific and clean binding profile. When we look at the product binding profile in intact cells, we can still see stabilization of the E3 ubiquitin ligase cereblon, but at the first glance, we don't see any CDKs. But if we look at the underlying data for the proteins, we can see what is called the hook effect. When the concentration of the product molecule increases to some level, then it prevents from forming ternary complexes and prevents the degradation. So what we see is the protein level first goes down and then goes up, showing the stabilization of the CDK. And this hook effect we see for both CDK6, CDK4, but we also can see it for off-target kinase, casein kinase RA. What is also interesting here is that the destabilization of RB1 happens exactly the same way with both treating cells with palbociclib and the palbociclib-based product molecule. All these molecules cause exactly the same destabilization of PLK1 kinase. Just to summarize this part, we can conclude that simultaneous measurement of the protein stability and abundance alteration brings together target engagement and pharmacological activity. And CETSA Explore with mass spectrometry readout can be applied for studying and developing of targeted protein degraders. And on this, I will leave the floor to my colleague Stina.
Thank you, Alexei. And I'm going to switch gears a little bit from small molecules and well, the focus on Alexei's talk on degraders into apply CETSA on therapeutic antibodies. So I'm presenting this on behalf of my colleague Verle Paternoster, who unfortunately she wasn't able to present today. So the work that I'm going to present is research project we're running in collaboration with our partners. So this is the project called PreBioBM, and it includes Pelago and then Pangea Oncology, and then NTRC, and it's founded by Vinnova and Eurostars. So in the case study included here, we have profiled Keytruda. And Keytruda acts by binding to PD-1, and by that inhibits the tumor deactivation mechanisms of the T cell. So going back to the Navigate format, so using Western blot, we have profiled Keytruda in PBMCs. These have been activated with the CD3 antibody. And then we incubated with Keytruda or a control human IgG. The incubation was for 60 minutes at 37 degrees. And then we applied the heat, so the heat ranging from 37 to 63 degrees. And the effect with Keytruda on PD-1 was a clear destabilization. So going from 39.7 TM, shifting no, starting from 50.8 TM with just the control, that was then lowered down to 49.7 TM with antibody. So this was also followed up by doing a dose response. So just as in the previous experiment, it was a pre-activation with the CD3. Same time for the incubation, so 60 minutes at 37 degrees. We did the heat shock now at 50 degrees. And clearly you can see it's a dose response on the effect on the thermal stability. And from this, we could also get the CETSA EC50 on the Keytruda antibody versus PD-1. An additional work that World has been working on is to apply the CETSA Explore format. So looking proteome-wide on the downstream effect of the initial binding to PD-1. So hopefully this is something that World will be able to later in the year come back and present to you. So stay tuned. One question that often comes up when we are talking about profiling therapeutic antibodies with customers is whether the therapeutic antibody itself would melt. So this is something Verily has looked into closer. And in this melt and shift curves, we also included the amount of Keytruda across the different temperatures. And as you can see, there's no effect within this temperature range on the antibody itself. But actually looking closer at that, at the higher temperature, you can see the antibody itself starting to melt. And the melting is also affected on the buffer used. But as you can see, the temperature range was used in the Western blot format. Was up to 63 degrees, where we don't have an effect on the melting. And then with the Explore format, the mass spec format, we go up to 66 degrees, but then we don't see any melting in the CETSA buffer that is being used in those experiments. So the answer is yes, the therapeutic antibodies do melt, but in this case, it's outside the standard temperature range used in CETSA. So to conclude, you can use CETSA to confirm target engagement of therapeutic antibodies. And that is then in the CETSA Navigate format. So doing melt and shift curve experiment to confirm that. And then also you can also do a dose response determination to rank the different antibodies versus each other. And then you can also use the CETSA Explore to understand the mode of action of the antibody and the biology. And then last but not least is that the heat treatment has an effect on the therapeutic antibody, but it's out the temperature range used in typically in a CETSA experiment. So now we're coming to the end of this webinar, and I'm just going to summarize what the team here has presented to you. So the CETSA Navigate HT, as you saw in Helena's presentation, can be used for primary screening and thereby generating tool compounds, but also you can apply it for hit confirmation and generating SARs. It's a robust and sensitive assay, which allows for both high and low affinity binders to be identified. And as I showed you now, and also the data Benedict showed you earlier, the CETSA Navigate format can be used for confirming target engagement of both therapeutic antibodies as well as small molecules. But then also CETSA can be used for profiling degraders such as PROTACs. In that case, it can also be used to look at the target protein as well as the E3 ligase. And the CETSA Explore proteome-wide format can be used for studying both the direct binding events as well as downstream cellular pathway effects. And applications then range from selectivity studies, validating your tool compounds, benchmarking, and mode of action deconvolution. So thank you for listening. The next is questions. I saw earlier there were many questions that had come into the chat. So thank you all for being so engaged in this. The first question was regarding if there's no antibody available for detection. I guess this was early in the webinar before Alexei had presented the proteome-wide MS-based format. So yeah, of course you could study your compound using the CETSA Explore to identify targets. But also if you have a specific target of interest, but there's no antibody available, one can also consider maybe doing a targeted MS format, something we have started to look into at Pelago. Then it was a question regarding the hit rate in CETSA. And as Helena showed you, it's typically range from 0.2%, as in the case of STING, looking at the stabilizers, up to 1.2, which was the hit rate on stabilizer we saw in the CDK4 case. And then how to identify the false positives in the targeted CETSA format. So what we typically do is to change the order we're doing things in the assay. So for example, we would start with heating the cells and then adding the compound. That would be a way to see if there's any effect from the compound itself. As they were added after the heat shock, there wouldn't be an effect on the thermal stability, but more as the interference of the compounds. What is your criteria to pick hits? We typically use the free standard deviation as a cutoff to select hits to prosecute further. And related to that was a question if compound were binding both or showing effect both in the CDK4 as well as the STING, if they would then be non-specific compounds. And that could be one reason, but it could also be that they are interfering with the assay readout. So the reagents, the detection reagents. Let's see. And question here whether it's on the benefit of profiling degraders in the CETSA MS format versus just a standard global proteomic study. I would say that in a global proteomic study, you would understand the degradation specificity of your degrader, which of course is very important. But then in addition, if you do a CETSA study, you would also understand what else your compound is interacting with. So what is the degrader interacting with? Not necessarily degrading those proteins, but potentially inhibit them or affecting them. So off targets of the degraders. Then there's a question, what is your criteria choosing cell lines? Well, this depends on your biology. We would recommend that you stay in a cell line where you have the relevant biology. But then, of course, if it's a screen, it's something also that needs to be able to, it's a cell line that needs to be able to be cultivated as we need to get the cell numbers up. Yes. See here, we can take one. What is the percentage overlap of hit rate in intact cell versus lysate? I think that is very compound dependent and probably also a lot of target dependent. We know that some targets are mainly working in the intact cell. So that could be a difference between the lysate and the intact cell in that case. And also, some compounds are more active in intact cell setting than in a lysate. And vice versa as well. If you have compounds that are not cell permeable, you will see an effect in the lysate, but potentially not in the intact cell experiment. Yeah, I think we could, we can take this last question here. How do you select the temperature you used for normalizing? So typically we use the 37 data point for normalizing, and specific for the data that Benedict showed, I will leave it for him to follow that up. Yeah. Well, the rest of the questions we will follow up in emails to you. And I always like to end with thanking you all for your kind attention. And yeah, thank you for joining us now, and bye from Stockholm.
These people are scientists, directors, and portfolio managers. This guy is a drug discovery project leader. They have one thing in common. They all failed to check relevant target engagement early in their projects, so they can't see where they're heading. That's risky. Ouch. Let's make sure it doesn't happen in your project.